Okay, hi everyone. So today we will have another lecture. Okay, so following on our previous lecture, we have learned about the UV visible spectroscopy, and then you have learned about each of the instrumental part in a UV visible spectroscopy. So now when you look at this slide, it shows that a plot of absorbance versus wavelength. So as I mentioned before, this is a typical UV visible spectrum. Okay, so as you can see that from this graph or from this spectrum, it shows that whenever that we have an increase of concentration, the absorbance also will increase. So as what I've mentioned in Beer's law. All right, following on that is that we are going to look at deviations, or we can say that errors that can occur in UV visible spectroscopy. So the ideal output from UV visible spectroscopy is B, means that it's following Beer's law with no deviations from Beer's law. But also during experiments, we also notice that there are going to be some positive deviations and also some negative deviations, okay, as mentioned and as shown in the graph. So the ideal way is that B, no deviation, but we also would predict that sometimes it can give positive deviations or negative deviations. All right, so what causes these deviations actually? All right, causes of deviations from Beer's law is that there are going to be two, which is instrument limitations and as well sample limitations. So what are the instruments limitations here? You can see that listed is resolution, signal to noise ratio, stray light, and contamination. So all of this comes from the instrument itself. So instrumental error lah, something that we might say. Instrumental error. How about sample limitations, molecular interactions, associations, hydrogen bonding between the molecules, the reactions that causes by the molecules, and also the refractive index changes. So all of this would create some kind of limitations either by positive deviations or negative deviations. Okay, so the third part is the chemical deviations. So when we look at chemical deviations, it changes in concentrations. Okay, sometimes you can get a concentration. Okay, for example, you have standards. You have standards where you prepare only 100 milligram per liter, which is 100 ppm. 100 ppm apa of standards. So when you do your calibration curve and when you do your analysis, you should get this 100 ppm back because you already prepared 100 ppm. Okay. Okay. Sorry. Sorry. Something happened to my iPad. Let me go back. I need to unshare this first. Okay. Let me continue. So basically for chemical deviations, you can see that there is a concentration that's going to change. And then these concentration changes arise when the analytes associates, and reacts with each other, either with each other or with the solvents to produce a product or also all of these reactions, dissociations, associations would create some kind of deviations, would create some kind of concentration change. All right. So, for example, when we looked at associations, for example, for matrix and analyte, you can see that it's causing original analyte to be less. Okay, or even the interaction between matrix and matrices form a compound that absorb the same wavelength as the analyte. So all of this will contribute to the concentration changes. Sorry, I'm not sure what happened, but never mind, we will continue. Okay, maybe just a little bit more on this. All right, so the deviations you will understand, okay? We have different kind of deviations and what it causes uh, to your analysis. Okay, so this is there a question here. Two different sample cell can be used in a measure. Can be used to measure. Can two different sample cell be using to measure? So can this be allowed? The answer is no, because as I mentioned before, you need to use the same cuvette to measure your sample and also your background, because this is to minimize the error. All right. So now we are going to look at on the applications of UV visible. There are a lot of applications here. Vitamins, analysis of fatty acid, analysis of proteins, trace metals, and food coloring. So maybe we can look at one of them. So analysis of analgesic tablets. So this is a paracetamol. Paracetamol. This is paracetamol punya structure. So basically, when you looked at on the type of sample that can be used in UAVs, you can see that all of these are molecular substances. Okay, so such as paracetamol. So this is a paracetamol molecule. How about this one for food coloring? You can see that this is a structure for catechins. So all of these are molecular, because after this you are going to learn about atomic spectroscopy. Okay. All right, so maybe I should just finish off a little bit more before the iPad makes it worse. All right. Um, so as you know that UV visible is quite universal in terms of um, having a lot of samples that can be analyzed by it. So yes, as we looked at on the catechins, so this is the catechins, and then also towards the end of the slides, you can see the analysis of synthetic colors also. All right, so for the application, I think that you can read it on your own, because I think that is quite straightforward in terms of how to prepare, and then what about the results? Okay. All right. Thank you.